So good afternoon to everybody, and we go ahead with the second monitoring section. I will remind you that you have 10 minutes for your oral presentation, and then we will leave 5 minutes for the discussion or the question. So the first speaker is Roberta Ferrante. So welcome, Roberta.
Good evening, everyone. I would like to thank the committee for selecting my abstract as an oral presentation. I'm Roberta Ferrante, and I'm a PhD student at the University of Florence. And today I will talk about my PhD project titled Developing a Comprehensive Biodiversity Index for Sustainable Forest Ecosystem Management. In the recent years, we have been observing a biodiversity loss at global, regional, national, and local levels. The main forces of biodiversity loss are climate change and anthropogenic activities. The genetic resources and biodiversity that characterize forest ecosystem must be conserved. Genetic diversity is one of the 3 components of biodiversity, and now it is declining. Genetic diversity underpins evolutionary change and plays a key role in response to climate change, habitat alteration, and biotic interaction. So, low genetic diversity increases the risk of extinctions. So far, biodiversity has been discussed at species and ecosystem level, while genetic diversity is often not incorporated in this discussion, despite its importance. So, to monitor the loss of biodiversity, specifically genetic diversity, and to understand how biodiversity can support ecosystem response to climate change and human activities, it is important to include genetic diversity and to implement a genetic monitoring program for effective forest management and conservation. The goal of my PhD project is to develop a comprehensive index for quantify biodiversity in forest ecosystem, to preserve and to monitor biodiversity. Moreover, we want to identify the best silviculture practice that improves forest biodiversity, which is a powerful tool in response to climate change. To achieve this goal, we analyzed 8 forest species presented in 31 demonstration sites located in Italy, Slovenia, and Croatia. We collected genetic, forest structure, deadwood, soil, and macrohabitat data in order to calculate verificators used to define 5 types of indicators, namely genetic indicator, forest structure indicator, deadwood indicator, soil diversity indicator, and microhabitat indicator. For genetic indicator, we analyzed nuclear macrosatellite data and single nucleotide polymorphism data in order to study neutral and adaptive genetic diversity. As preliminary results, we studied 4 forest species. For Abies alba, we analyzed 6 sites, for Fagus sylvatica, 12 sites, for Pinus pinea, 3 sites, as well as Quercus robur. In these slides, I reported a small portion of a data set that we created in order to collect all biodiversity indicators that we calculated. Moreover, here I reported an example of indicator for each type of indicator. In a total, we calculated 51 indicators for each species for each site. Regarding the results obtained for Fagus sylvatica, we observed that all growth forests showed the highest biodiversity level. However, across managed sites managed according to individual tree selective thinning, showed similar level of biodiversity found in all growth forest. In these slides, I reported an example of sites managed according to this type of silviculture practice. In these two images, we can see that the stands exhibited a complex and heterogeneous spatial genetic structure. In fact, we can identify 4 clusters. Moreover, by analyzing forest structure, we can see through these plots that the stand is characterized by a J-shaped curve of the geometric cluster distribution. And regarding the vertical structure, we can see that this stand exhibited a multi-layered pattern. As preliminary results, we created a synthetic index by selecting key indicators that indirectly describe genetic diversity and represent biodiversity in terms of deadwood macrohabitat and species diversity. Regarding the results obtained for Abyss alba, we achieved results similar to those found in beach stands. In fact, we observed that all growth forests showed the highest level of biodiversity. Also in this case, the management practice that maintain high level of biodiversity is individual tree selective thinning. Also in this case, I reported an example of a site managed according to this type of silviculture practice. In fact, we can observe a complex heterogeneous spatial genetic structure, and we can identify 6 clusters. And moreover, by analyzing forest structure, we can see through these plots that the site presented a J-shaped curve of geometrical class distribution and a multi-stratified pattern. Also in this case, we created a synthetic index for describing the biodiversity present in the stands that we analyzed. For Quercus robur, we found also in this case that all growth forests show the highest biodiversity level with high genetic diversity and a complex spatial genetic structure. When these stands that we analyzed are not pure stands but are mixed stands. So when we analyzed all species present in the stands, we can observe a J-shaped curve and a multi-layered pattern. However, when we observe only Quercus robur, we obtained a bell-shaped curve and a monostratified plot. Also for Quercus robur, we created a synthetic index by selecting key indicators for describing biodiversity. Regarding Pinus pinea, we found a low genetic diversity with a simple spatial genetic structure. In fact, in this slide, I reported an example, and there is only 2 clusters that testify a simplified spatial genetic structure. However, when we observe forest structure, we can observe a J-shaped curve diametrical class distribution and a multi-layered pattern. So, based on our preliminary results, we assume that the effective management approach that enhances forest complexity by creating a multi-layered structure which facilitates pollen dispersal, promotes genetic diversity, and increases new allelic variants crucial for climate change adaptation. Moreover, our next step involves the correlation of the identified indicators for deeper insight. Thank you for the attention.
Thank you, Roberta. Time for questions.
Hello, I'm Giovanni Felici, Spoke 7, University of Padova. I would like to know if you know, by chance, an indicator, an index that use satellite images for giving an indicator, a measure of the biodiversity status. So if it is possible to to calculate the biodiversity status from a satellite image? I know the end why, but...
Thank you for the question. And I don't know. It's not my field of research. So.
Hi, thank you for your presentation. I just wanted to ask, I didn't understand how you calculate the biodiversity index, how you get the synthetic index you...
Yes, thank you for the question. And we are working on that. We have a preliminary selecting key indicators in terms of information regarding forest structure, because we observed that multi-layered pattern of a stand promotes pollen dispersal because there is less barrier for pollen dispersal, so improved genetic diversity. For now, we are decided only these indicators, but we want to correlate genetic indicators and forest structure indicator in order to conclude the biodiversity question.
Hello, thank you very much for your talk. And I just wanted to know, because biodiversity is an important factor, but it is, at least to my knowledge, not the, for example, the species richness. It's not that an ecosystem is more stable, is better just because there are more species. They have to be a certain type of species, so that must be a role of each species. Do you plan to develop an index that takes into account this?
For a short time I didn't deeper all the indicators and all the verificators, but we include also speech richness and evenness, and but we focus on indicators regarding genetic diversity, because there is a knowledge gap and we want to fill this gap.
So Roberta, just a curiosity, how did you choose the species?
It's part of the project where I'm involved, and they selected this species before in this project. But
Okay.
they selected this species for monitoring. They want to observe how silviculture practice impacts on biodiversity and specifically in genetic diversity. So we would like to identify the best silviculture practice that maintain high level of biodiversity. And for this reason, we choose this species that's important at economic level and for biodiversity.
Okay, thank you. So time for the next speaker, that is Francesco Ferraro, that will present to us the advances in the modeling of ecological communities with a theoretical physical approach.
Okay. Hello, everyone. It's very nice to be here. I'm Francesco Ferraro at the University of Padova. I'm a physicist. And today I will talk about advances in the modeling of ecological communities from a theoretical physics point of view. So first of all, let me highlight two words in my title that I think most of you didn't expect to see today, which are ecology and physics. And many of you might be wondering, what does physics have to do with ecology? So let me start by telling you what is physics. Physics is the study of nature. And the grand goal of physics is to find the laws that are present in nature and use these laws to explain the regularities that are present in nature. And what I mean by regularities? The regularities that physics is interested in are present in two kinds of systems, very broadly speaking. The first kind of system in which these regularities appear are systems in which there are very few objects that interact. As an example, take the solar system. The solar system is composed of the sun and 8 planets. So there are few objects interacting. So it's no surprise that it's very regular. And it is very regular, for example, by the laws of Kepler. Like the motion of planets is not random, but is on a circle or an ellipse in general, in which the sun has a specific position. So it is no surprise that it has a lot of regularities. Another field in which there are a lot of regularities is particle physics. Particle physics essentially deals with interactions between pairs of particles. Like, for example, in this diagram, you're looking at the interaction between an electron and a muon. So it's no wonder that when you have very few degrees of freedom, as we would call them, then you have a lot of regularities. And these regularities in particle physics are explained by the standard model of particle physics, which is maybe too good at explaining these regularities, but that is another story. But you may not know that these kind of regularities appear in another kind of system. And these are systems in which the objects interacting are in the opposite limit, in which there are many objects interacting. Like, for example, gases. Gases are made of billions and billions of molecules, and the fact that you have so many things interacting, in a sense that can be made precise in physics, all the details start to don't matter anymore. Like, for example, in gases, you have the gas law that relates the pressure, the volume, and the temperature. So when you have many things interacting, then you find regularities. Another kind of systems that you may be familiar with is flocks, like bird flocks. And this was, you may know because this was the subject of study, one of the many subjects of study of Giorgio Parisi, the recent Nobel Prize winner. And the motion of a single bird in a flock is very complicated, but when you have lots of birds, then regularities start to emerge, and you see these beautiful patterns that you observe in nature. So you might start to understand where I'm going, because ecological communities are composed of many species, many individuals, and so they present regularities too. And just to give you an example of the kind of regularities that, as a physicist, I'm interested in, this might be a bit complicated at first, but these are data collected from bacterial communities, which were compiled by this very good theoretical physicist, which is Jacopo Grilli in Trieste. And he noticed that some macroecological patterns are very regular in different communities. For example, here is data taken from bacteria in a glacier, in a human gut, in lake, rivers, seawaters, and so on. And for example, if you look at these IFDs, a microecological pattern which basically tells you what is the fluctuation in the species of mundaces, if you rescale the things properly, what you find is that all the data from very different bacterial community collapse on a single curve, which is a gamma function, but it's not very important. So there are regularities, and this is just one example of a regularity. You can find also regularities if you average in some way that I will not talk about, you find that all the data collapses on precise curves. So there must be some laws that is governing the behavior of this kind of systems, which give rise to these regularities. So it's important to have a model for ecological communities. First of all, because if you have a model, then you can uncover the principles and the laws that are behind this kind of behavior. And once you have these principles and laws, you can explain the regularities. Because physics is not only interested in understanding why, understanding that there are regularities, it also wants to understand why. And why do we want to understand the regularities? Because then you can make sense of the data that you are collecting. Because otherwise, if you take data from an ecological community, and if you don't have a model that allows you to connect the dots, then all the dots are more or less incomprehensible. And if you have a model, you can fit the data to it and make sense of what you're observing. And once you have a model, and you have the data to fit the model, then what you do is to make predictions. Take the solar system. Once you know how gravity works, and you know the position of the planets, then you can predict where the planets will be in the future. And the same thing is true in ecological communities. Once you have a model that you can fit with the data, then you can predict where it will be going. So it's very important in application to have these kind of models because you can predict what will happen. And moreover, you can also enable the control of these communities. Just think of what could be enabled in applications if you could control the bacteria that live in your gut. You can move them from an unhealthy state to an healthy state if you have the model and you have the data on which to fit the model on. And just to give you a very little taste, to be more concrete on what we do, I will present you a classical result on the stability of ecological communities. And what I mean by stability? By stability, I mean take an ecological community at equilibrium. What does it mean to be at equilibrium? It means that the number of individuals of each species is not changing in time. It is more or less stable. Then what you do is you make a small perturbation, okay? What does it mean? It means you remove some individuals of one species, or you remove a species of many species. And if it returns to the previous equilibrium, then you say that the system is stable. And if it does not, then you say that the equilibrium is not stable. And this is basically the definition of stability. And a very, very important result was obtained by Robert May in 1972. Robert May was a very influential physicist and ecologist. He showed that a community, an ecological community, will be stable if this condition is satisfied. So he made a quantitative prediction of the stability of ecological communities, where in this formula, the sigma is a measure of the diversity in your ecosystem, S is the number of species, and C is the connectance, so how many species does each species interact with. And this result might be very surprising to you, because it's telling you that the more diverse, so the larger the sigma, or the more species you have in an ecosystem, or the more connected it is, the less stable it is. And this is very counterintuitive, because as many talks have told you yesterday and also today, what you expect is that the biodiversity should increase the stability of ecosystems. And this paradox, which is called the complexity stability paradox, is still unresolved 50 years later. So still, we are lacking a quantitative framework which is able to explain the patterns that we observe in nature, in the ecological communities. So that is what I'm working on, towards developing such a model that is able to describe ecological communities. And I want to tell you more in detail what I'm working on. And what I'm working on is the so-called Lotka-Volterra equations of community ecology that basically say that the growth rate of a species, here appears a derivative, which means just the change in time of the number of individuals n of species I, is proportional to the individuals that are present in the community. And here I'm adding a little bit of complexity, since this G is not constant, but is a function of the whole community. And this is what I'm working on, to study the Lotka-Volterra equations and to understand if they can describe ecological communities. And I won't go into detail with this. And I want to tell you what we have been doing. I work at the laboratory of interdisciplinary physics at the University of Padova. And our efforts have been, first of all, to show that if you take the Lotka-Volterra equations and you allow for interactions between the species to be dynamic and not static, then what you observe is that you can derive some more realistic microecological patterns. Moreover, what we showed was that, what we showed that if the interactions between the species are not instantaneous, but they are delayed, then in your species abundances, it will appear persistent and synchronized oscillations. Also, as I told you before, some patterns in ecological communities are universal. They don't depend on the specifics of the ecological community, but some of them are not universal. And we were able to give an explanation of the origin of this non-universality. And now we are doing more work to bridge these kind of abstract models to the observations that you observe in the most popular experimental communities, which are bacteria, forest, and plankton. And also, I invite you to check out Alice Doimo posters, which goes into more details on some of the work that we are doing. If you want to know really what we are doing, it's quite technical, but you can sense what we do. And to conclude, what I want you to get away from this presentation is that communities, ecological communities, are the simplest level of collective organizations in ecosystems. And ideas and tools from mathematical tools from theoretical physics can be used to study them. And if this interested you, I'm very happy to talk about it in more detail. And my group is very eager to collaborate, especially in connection with data. And my specialties, if you're interested, are mathematical modeling, simulations, and data analysis. So if you think that there is a connection between my work and what you are working on, please get in touch. I'm very happy. And I want to thank you for your attention. I will take gladly any question.
Thank you, Francesco. Questions?
Thank you, Francesco. Really nice talk. Also, I really appreciate the format you used. And maybe it's something more philosophical, let's say, but I think also philosophy is another word that has to be linked to ecology. And before you talk about starting from a stable situation and reaching another stable situation. And I'm wondering if we can really obtain or we can really have an ecosystem that is not experiencing stresses from the outside, so that can be really stable. If I mean, maybe should we just think that ecosystem pass from one stable point to another one because they are always influenced by the external forces?
Yeah, thank you very much for your question. Actually, this is one of the critiques of the model that was used by Robert May in deriving this result. And yeah, it does assume that the equilibrium is stable. And this stable point is not changing in time. So that is a very strong assumption. And it's very interesting that you mentioned this, because right now we are working in extending this result to the case in which the equilibrium moves, which is what happens in reality. So I can keep you updated with that, but that's a very good critique of the thing.
Yes, my question was similar to this one. I would like to ask you if you will take in consideration your model also time, because the stability.
Okay. Yeah, sure.
I mean, in presenting, you say to perturb slightly,
Yeah.
and say maybe if we take out a species, but then to recover, it takes time. I mean, that was the basis also of the Loft-Volterra law. I mean, they start from, in particular Volterra, from the fishing data after the war. So it took time, to recover or to re-have equilibrium from predator to prey. So maybe a model could be perfect in just giving you a picture in this moment, a fixed one, no? But if we want also to use as a tool to predict, maybe the temporal aspect could be important. I know that it could be complex, but...
Yeah. Actually, if you look at the model of Robert May, it does include time in a sense, but not really, because it only deals with a fixed situation. But the moment you go to the Lotka-Volterra equations, here I'm not saying it, but here, you see, is a T of time. So the Lotka-Volterra equations are intrinsically dynamical. And I didn't show you, but these are the kind of trajectories that you see in the model. You like, for example, you start from a random configuration and you see that it evolves in time. And depending on the parameters of the interaction, this evolution can be towards an equilibrium. As you can see, these lines are reaching a stable point. Or they can fluctuate in time, which is maybe more realistic of your ecosystem. But let's say that the Locked-Voltaire equation are the next step of the model by Robert May, because they include the dynamics also, not only
the temporal scale.
Yeah, exactly. Yeah.
So actually, the data that you are using to test the model, you say that you are using data on bacteria, on forest. Do they come from, where do they come from, these data?
Well, that's a very tough question, because I usually look at databases which aggregate a lot of, like, for example, there is a very big database, it's called DPG, I think, which has a lot of time series, which you can fit the Lotka-Voltaire equation on and understand what are the parameters in your model. And for bacteria, actually, I don't know where Jacopo Grilli, which is basically the spearheader of this approach of physics into microbiology more than ecology, I think just contacted experimentalists.
Maybe the data that your colleagues are collecting.
Yeah, sure.
Could be really
that's what I'm
important.
saying. That any kind of data is useful to us. So if you think we have a model that
it could be a really good example of translational science among PhD students.
May I ask something?
Yeah,
sure.
Yes, please.
So you show the regularities plot on microbial communities.
Yeah, let me go.
So I was wondering if that regularities that we see, isn't that indeed the results of the way that we measure microbial communities, because we know that we cannot see the whole picture of microbial communities when we sample them. So I was wondering if those regularities reflect the biases of the method that we have to measure them.
Okay, I'm not very expert on the way the data was collected and sampled and so on, but you see, if there was no regularity, then this data would not be on this curve. Like, this is not a byproduct of, I think what is happening here, they're just looking at time series and looking at the fluctuations in the species abundances of each species, and then collapsing them on. Of course, you cannot just take the data, you have to analyze it in the correct way. If you do it the correct way, the data points don't look scattered, but they lie on we would say on a lower manifold. So they are on a specific line, and that means that if you take an ecological community for this one, and you want to know what is the fluctuations, then you can use very general models without having to specify the specific ecological community. But I don't think, I know Jacopo is very, I don't think that there is something fishy going on. I mean, these plots are very telling of what is going on. There are regularities both in the abundance fluctuation distribution, the mean and the abundance distributions, and also in this direction with Taylor law, and they are very fitting, I would say. So yeah, it's not a byproduct of...
Thank you.
Yeah. So thank you. Thank you, Francesco. And now we have Irene Gregory speaking again on microbes, microbial responses in Venetian lagoon, salt marshes, dialing sites. Go ahead, please. Okay.
Hello, everyone. I am Irene, and I'm working in the microbial community within the Spoke one, so the marine one. And what I do is to use molecular methods to characterize the microbial community, to try to understand how they interact with the ecosystem functions, and then monitoring the changes over time. I'm doing this in different coastal lagoons ecosystems, but today I'm going to talk about my favorite experiment, which have been done in salt marshes. So they are complex ecosystems found in the interdial zones of the estuaries, and so they have been submerged when the tide is high, and they go out of the water when the tide is low, basically. For those of you who went in the salt marshes, you know that they not look like this in reality, but they are really muddy. And still, they are very important habitat for many different species. For example, they are a nursery ground for many fishes and avian species. They have an high organic components, especially in their sediment. They act as a buffer against coastal erosion, and they act also as important carbon sinks. We don't know much about the ecology and the microbial ecology of that environment. Because they need to be preserved, we have to understand how they work, and is what I'm trying to do. I'm doing this basically with molecular methods. So this is the methods I'm currently using, simple metabarcoding. So I'm collecting some environmental samples. I'm extracting both the DNA and the RNA. I'm amplifying this with specific primers through simply a PCR. And then I analyze the results of an high-throughput sequencing with a bioinformatic pipeline to have a result, a list of taxa that I found in the environment and the abundances of them. So what we did was to choose 2 salt marshes in the southern basin of the Venice Lagoon, mainly for logistic reasons, because they are near the land and they are connected between each other. We choose 1 natural salt marshes. Basically, we were camping there. We went to the natural salt marshes, and then we work in the artificial one. We choose to characterize the community over 1 DL cycles because we know that there are many shifting in nutrients and some parameters over the DL cycle. So we choose to sample over all of that. And because it was very hard, we collect as much as we could. So we collect 24 sediment samples for the NMR, 20 water samples for many nutrients analysis, and also the 24 sediment samples for my meta-analysis, so metabarcoding, and immediately frozen in dry ice. We also collected parameters using a mini dot logger, which is this sensor, which is able to collect parameters such as temperature and dissolved oxygen in continuous, so like every 1 minute, 2 minutes. And we put this in the water between the 2 salt marshes. And also, each time we went sampling, we use a multiparametric probe to validate the collection of the parameters also right in the site of sampling, basically. So these are the results of the mini dot. We can observe. We let it be in the water for 3 days, and we can observe very high, strong fluctuations of temperature in black and dissolved oxygen in red. And also with the multiparametric probe, we observed a shifting of 10 degrees in the temperature of the water, and a shifting from the 20% to the 86% of dissolved oxygen between the night and the peak during the day. So starting from that, for example, for the nutrients, we observe there are a pattern in many of them, with the phosphate, for example, increasing during the day and the ammonia increasing during the night. And also, we found the differences between the artificial and the natural salt marshes. So we did the metabarcoding, analyzing the V4 and V5 hypervariable regions of the 16S. We successfully characterized the microbial community in this way, finding about 400 taxa. The majority of them were in the Bacteroidales order, Cytophagales or Flavobacteriales, which are all involved in the metabolisms in marine communities. But unfortunately, there were no differences in terms of community compositions between the samples over the DL cycle. At that moment, it was a little bit sad, but then it makes sense because the sediments are very stable. I was expecting some differences because I read all about water community compositions, and they change a lot between the DL cycles. But apparently, the sediment is not the same. Starting from this, we also characterize the eukaryotic community, amplifying the V9 hypervariable region of the 18S. We found that the most represented phyla were Arthropoda, which is this one, and Cnidaria and Diatomea. And even if we see some differences between the DL cycle, the majority of the difference was comparing the natural and the restored salt marshes. For example, Cnidaria are only found in the restored one, while Annelida only in the natural one. So what I'm going to do next is maybe the metagenomics, for sure the metatranscriptomics. I'm starting this by extracting also the RNA and to sequence the RNA with the aim to obtain some information about the expressed genes in the different environmental samples, with the final aim to understand the ecosystem functions of each of the taxa present, and hopefully I will observe shifting in the functions of the same taxa during the day and during the night. So, thank you.
Question?
Thank you for your presentation. That was very interesting. And I would like to know more about the differences between the restored salt marshes and the natural one, and what is your, why do you think it's, for example, you observed a different composition of taxa, especially for the last part you showed?
Yes. So what we, after the results of the... Posso, prof?
Ma certo.
No.
Sure.
Okay. So after the result of the eukaryotic metabarcoding, I was questioning myself, why is the Cnidaria is all in the artificial one and the Arthropoda are all in the natural one? I don't explain myself with the granulometry, which I'm still waiting. I'm just seeing the results, but don't make sense. I explain myself because of the position of the two. So this is the natural one and is closer to the land, while the artificial one were closer to the sea. So I expect, for example, the Cnidaria, which have the moment in their life they have to be bentonic, to be all in the tidal channels that we sample, but more closer to the sea. While Arthropoda, maybe they prefer a more dry land, for example. This is why I'm expecting this. While for the bacteria, there were no differences. So.
Irene, if I'm right, you sampled only in late June?
Sì.
Okay. Do you intend also to sample in other season? Because, you know, don't remember, but you sampled in 2-3 days.
Yes, in 2 days. It was enough.
2 days, okay, 2 days. So honestly, I will never expect to find variation in the community in 2 days, 6 months.
No.
So I'm asking if you intend to sample also in other seasons.
I don't know. We are thinking about it, but we also choose the timing in which there were more shifting in nutrients and in the parameters. So if I don't find differences in this, I won't expect to find them in winter. But it's something we could do. It's something we can also do is to try to see there are some differences between the two salt marshes. For example, one which have been restored earlier in respect than this one. Or this is the open question. But it was really hard and time consuming.
No, I can imagine. I can imagine. I don't want to discuss about. It's just because, of course, in winter you have the amount of nutrients and other parameters that are different. So maybe they put selective pressure on some of the taxa.
If we can, we will, for sure.
Let's hope you will have the possibility to.
Thank you.
Hi, Irene. Thank you for your presentation. I'm just wondering if maybe for future challenges, do you want also to study the viral community interaction with the bacteria? For instance, if you expecting that some auxiliary metabolic genes can alter the stability of your bacteria community or something like this?
Actually, a colleague of mine, which is not here, unfortunately, is doing that. So we went sample together. We have the same samples, and we are sharing them. And he's analyzing all the viromics part. Including the metagenomics, so all the bacteria, the archaea, everything, and coupling them with the viromics part. So we are doing this. Thanks.
One quick question. Do you expect the artificial salt marsh to become more and more similar to the natural one in terms of biological composition and other parameters over time, over a long period of time?
Yes, I expect this. That's why we started with the salt marshes that had been restored 34 years ago, so many years ago. And also, they are, in this case, they are colligated. Basically, there were, at one point, the natural became artificial because they attached a part of the salt marshes to the natural one. So I expect in this case that there are less differences compared to one in just restored 5 years ago. That's why we are doing this, because we were thinking about collect as much information as we could in order to, I don't know, make the microbiota more similar just from the beginning, from when we are restoring it, trying to make them easily similar with the natural one.
At the same time, you know, the position could be crucial because the natural one are close to the land, the other one close to the sea. So maybe you can expect that in time the similarity increase, but still the effect of the position could be important in making the two communities slightly different. Other question? So we can go ahead with the... Thank you, Rainer. With Roberto Magnolini, the last one, that he will present us the biodiversity of neglected animals. So I'm quite proud that somebody talk about, minorities, and so maybe this taxa is not a minority, but is for sure neglected. So yes. Let's see what Roberto will tell us.
Okay.
Hello, everybody.
I hate centipedes.
Me too. um,
You have a good reason for
okay, I want to study biodiversity of neglected animals, in particular taxonomy and distribution of lineages of soil centipedes. It doesn't work.
No. Vediamo. Try again. Nobody wants to talk about it. It's a really a neglected tax.
Okay.
Okay.
So, as we know, there are a group of many group of organisms that are not adequately known and neglected, and we have a lot of evidence of that. And I concentrate in animals, but it concerns also plants and fungal. So, if we consider, for example, birds and the biodiversity occurrences recorded in GBIF, they are like 2/3 of the entire amount of the biodiversity occurrences, so 10 millions on 15 millions. While if we consider insects, for example, they are only 1 million, while if we consider the total amount of number of species, they are more than 1 million, while birds are about 11,000, I think. So this is what we call taxonomical bias, and it concerns also centipedes, of course. And focusing on my groups of interest that are soil centipedes, there are about 1,030 valid species, while the named species are 1,700. While considering Europe and Italy, in particular Italy, that is probably the most studied country considering the taxonomy, we have 70 species, but 1/3 is of uncertain validity. Uncertain validity considering morphology. If we will consider genetics, probably this number will increase. So I want to concentrate my study on a group of centipedes, Henia, that is a genus, and considering inside the Gnatodontini, and that lives in West Palearctic, especially around the Mediterranean basin. They are about 20 species, and they show remarkable evolution in ecology. As a matter of fact, some species live just in the mountains, in woods, while other species, they are living by the sea. And this is the history, the taxonomical history, considering the number of taxa that were included in Henia, starting from the first one that was found, and nowadays. So there is an increasing of the number of descriptions, while the number of species decreased because a lot of them were considered as synonyms. But if we consider this is the last article work that works especially on Henia, and so this and this decrease are just because there were articles and works on more general groups. So this is an unstable taxonomy, and the aim of my work is to make this group stable on a taxonomic point of view. So make a taxonomic revision of Henia and its relatives, considering geographical and morphological data. And of course, a second aim is to using morphological molecular data to make a phylogenetic reconstruction that could also help in the taxonomical revision, of course. And I perform a literature research considering all the description in history and also all the data about distribution. And also, I make specimen examination of a lot of collection. For example, there is a huge collection of centipedes here in Padua at the university. And also, for example, in the Museum of Bergamo or in Verona, and some centipedes were sent to me also by other private collection. So I go to the collections, I research them, and this is the result. So a morphological examination, for example, this is what we call a forcible. And talking about results, if we concentrate in Italy, there are at least 7 species, considering that I digitized about 200 of records, geographical records, and I examined more than 100 of species. This is an example of centipedes and a species that we can find in the mountains, in the Alps, and also in the Apennines. So, I want to make a couple of examples and of species, Enia brevis, Enia lyrica, Enia montana, and probably a potential new one that I found in the mountains, in the southwestern Prealps, and I know they are all quite the same. So let's go in detail a bit. This is just 4 examples I told about 7 species. That is just to show you that dimension can change from 2 centimeters to quite 6 in this species. Also, the leg bearing segments can vary from about 40 and to quite 90. But there are some species, for example, in Greece, that can reach about 150 pair of legs. So there is a variation also in the dorsal bands that can be absent or present. And also in the forcible variation, in the porphyry shapes, like an ellipsis, oval, circular, they call in literature biscuit shape, I don't know why, but also can change the number of articles of the ultimate legs. And to finish, I want to show the distribution. They are rogue data because, as you can see, we can say about, for example, Henia illyrica. You can see that is a species present in the Balkanic region, but probably these data are to be questioned. Also, for these species, this data probably is wrong and so on. So the next step of my work are to complete the literature records digitalization. Probably I have about 800-900 of estimated records that I have to digitize. Also, as I said, a critical evaluation of the records. I want to optimize the cladistic analysis because I don't show to you, but because it's very preliminary results. And then, of course, probably the most important part, but the most difficult part, starting the data collection, so the extraction, and so on, the analysis. And that's all.
Grazie. Thank you, Roberto. Questions? Could you please go back to slides? Okay.
Okay.
I'm wondering, was you that bring the lyric here?
No.
No. It's not. Because
These, as I said, sorry, maybe I was not so clear. So these data are not recorded by me, by myself, but are also from literature. So some, like these,
They seem
are
to
recorded.
be overplaced.
Yeah. And for example, these data, I know these data because are very strange. For example, they was collected by a specialist in the 1950, but the article was written post-mortem, so probably are wrong data and so on.
That's a tragedy.
Yes, it's very difficult. These are about 1,000, and probably there are more 100 of records that I have to digitize, and it's very difficult. It's like an historical work, and it's very time-consuming.
Roberto, I have a more serious question. I was joking, of course, asking if it was you that brought that sample there. Could you please go back again to the morphological traits, the previous one? So I'm wondering. Did you consider that differences in dorsal band could be a phenotypic effect? So, I mean, how you are sure that the criteria that you are using are good criteria to define the species?
Because most of them are quite stable, and of course the dorsal band are probably one of the most critical, because not just for the phenotypic variation that maybe is not so important because we can see that they are stable, but also for all the specimens that were collected in alcohol, the dorsal bands are not so visible, they disappear. So this trait is probably the more tricky one.
Okay.
Hey, thank you.
Excellent.
Yeah, thank you very much for the very interesting talk.
Thank you.
I'm just wondering. I mean, I think it's very interesting to study centipedes, and I'm sure that the reason for your interest is because they are neglected. But I wonder if there is some other reason because you are so interested in the taxonomy of these animals. Like, what kind of research question do you think studying these animals specifically you may answer in the future, if you have any? Or is it just driven by curiosity?
Okay. My interest in this type of animals, but not just centipedes, just neglected animals, is that if you have to answer to more specific questions, you have to have great taxonomy, are stable, and you have to define what you are studying. So we are doing this type of study also on other taxa, like for example some butterflies. And if you want to make some biological question more precise, you have to, in my opinion, you must have a good taxonomy. This is my point.
Okay.
Hello, I'm here.
Sorry.
I work on neglect taxa too, and I know
Which taxa?
Gastrotheca. And I know that there are a lot of issues caused by the lack of knowledge about the distribution or the lack of taxonomic keys and so on. So how do you face these issues in your study?
Considering, for example, I make some example, literature, we are very lucky because in my laboratory, Lucio Bonato, my supervisor, have the complete literature of all the history of centipedes. So it's pretty simple. Maybe the first step is to collect, and I know it's tricky to go in the libraries and so on, and collect a lot of old papers and old works, and have quite tall the literature of your taxa, and then if you want to work on taxonomy, of course. And then it's a good starting point. Without this point, a taxonomical work is a most impossible task. So the first thing is to collect more literature, and then start on the morphological collection we have in the museum, as I did. And this is the point, to have data to work.
Okay, thank you.
Roberto, I can imagine that it's quite important to have all the available records and literature. But the point that your fellow raised, it's important because you can have all the literature on this species, but this doesn't mean that you know about the distribution or the density, because maybe nobody took care about this aspect. So, I mean, do you intend also to make field collection by yourself, or do you just will use the preserved specimen? Excuse me. Basta. È testato, è stubborn. So do you intend also to collect?
Yes.
Okay.
Yes. Because the people that work with me has collected and are collecting now,
Okay.
and we are collecting a lot of groups of centipedes, not just Hynia, but a lot of Geophilomorpha. So if we want to perform, for example, DNA analysis, we have to have fresh material. So yes, we are.
So then you can have also some ecological information, where, when, in the litter.
Yes.
Okay.
Yes. And in my case, it's a hard task to have fresh material from Greece, from Caucasus, and so on. But I'm trying to to have it.
Okay. So thank you very much. No. There is another question.
I'm sorry to come back. Thank you again for your presentation. I'm
you.
just curious about something I saw on one of the slides. The time graph against the species, the one that went up and began to go down, you said it reduced because there were synonyms. Yes. Yes, this part.
No, sorry.
I didn't quite understand the synonym part of it. What do you mean by there were synonyms?
Okay, sorry. When you describe a species, and when I make an example to explain, maybe this year an expert make a description, and because the lack of communication, the next year another expert make another description of the same species, and then a third expert, maybe 20 years later, see that these two descriptions are of the same species. So this is a synonym. And this is why the curve decreases and the number of species, the number of taxa in this case, decrease. But it's because some experts noticed that some descriptions were of the same species and not of different 2 or 3 species, for example.
So does it mean there's a lack in, I don't know, a standard measure of the species? Because what is the probability that someone will look at yours now and say they are the same species in the future?
Sorry,
I don't know if there is a standard to measure the species. I'm just wondering that what if in the future somebody looks at your classification and say the 4 of what you've presented are the same species? Like, what is the standard to measure the difference between one species from another?
This difference is because, especially in the past, the description was a lot of morphological characters lacks in the description, and this is why we have different, the same species maybe, and it's difficult to, because there was not a standard in the past. Now it is, so it's more difficult now to make synonyms.
So thank you Roberto, thank you everybody. That's time for the poster session number 2, the coffee break. And then we will meet again all together also with the other group that is discussing about restoring. So maybe you have a round table all together. So enjoy the coffee and the poster.